Hello, can you hear me? Hello, can you hear me? Hello, can you hear me?
Yes, Lasha,
Okay.
but your voice is very low, Lasha.
See my PPT now? There's still 8 students and not in our class, but we will not wait for them, so we will start out today's class. Mm, Today we will talk about, you will learn about two experiments of the medical genetics. Um, because we have learned all the chapters of medical genetics of the theory class. In our theory class, um, let's review all these chapters. First is the introduction, um, then the classical genetics, and next is the gene and gene mutation. Then the gene mutation effect, cell effect, and then we learn about different kinds of genetic disease, the single gene disorder, polygenetic disorder, the population disorders, and then the mitochondrial disorder, and then the chromosome aberration. So from all these chapters, we can see because our class is medical genetics, gene disorder, polygene disorder, or mitochondrial disease, or chromosome aberration. But the fundamental of all these diseases is the gene mutation or the gene structure disorders. So, and the chromosome is the carrier of gene. So, in our experiment class, we will learn chromosomes with a specimen, then to observe its aberration. And as we talk about in our single gene disorder, effector, factors, it has the X chromosome inactivation, or later in the chromosome class, we say the male has only one X chromosome, chromosome X, but the female has two chromosome X. So, but the gene production for female is not more double than male. Why? Just because during the embryo development, one X chromosome will be inactivated, randomly inactivation. So, because this X chromosome is inactivated, so it is a kind of a heterochromatin. And then the heterochromatin can be dyed with can be dyed with some DNA dye, for example, the giemsa or the DAPI, or the giemsa or some other nuclear staining solution. So this inactivated X chromosome can be deep dyed. Then we can observe it under a microscope. So our first experiment is the observation of Barr's body in human cells. Experiment, you should learn and master the technology of Barr's body preparation from human oral. And then you should know how to observe the Barr bodies and a microscope with a Barr body specimen. Then you should understand the relationship between the number of Barr bodies and human gender. Barr bodies and human gender deformity. And then you should understand the measurement compensation effect. First, what is a Barr body? Because we should learn to observe the Barr body. But what is a Barr body? The Barr body is one inactivated X chromosomes of a woman. Women with two X chromosomes express as much genetic materials as men with only one X chromosome. It is a common phenomenon. But why? So later, the Lin provide a theory, a hypothesis, the Lin hypothesis, also known as the Lin's dosage composition effect. One of the two female X chromosomes was randomly inactivated at the early stage is activated and the other is inactivated. After we will learn the dosage composition effect. It is a genetic effect of the convergence of gene expression between individuals with two or more copies and individuals with only one copy. In human or in mammals, the dosage composition effect is a random inactivation of the X chromosome. But not all the species is the same way to ensure the same genetic materials between male and female. For example, in the marsupials, it is the paternal X chromosome inactivation. So it is the X chromosome that from in the female is the in the marsupials is the X chromosome from his father that is inactivated. And in the dolphin, in the Drosophila, it's not the female, but the male one. The X chromosome of the male one is present doubles. So there is genetic materials can be the same between female and male. And in the elegance, in females, the gene expression of both X chromosomes is halved. So the total genetic material is the same between male and female. Then next we will learn the discovery of Barr body. In 1949, Barr discovered a kind of condensed corpuscle called the Barr. A condensed bodies called the Barr corpuscle in female cat neurons, and the male cat did not have this condensed body. And in 1955, Barr discovered Barr bodies in female oral cells. Then, in 1959, malformations caused by sex chromosome abnormalities were linked to the study of Barr body. In 1971, Barr's body was called X chromatin. So here the down picture shows the Barr body. It is the condensed the number of the Barr body equals n minus 1. N is the number of X chromosomes. So, because male has only one X chromosome, so they have zero Barr bodies, no Barr bodies. And the female has two X chromosomes, so they have 1. 2 minus 1. They have 1 Barr bodies. And in our chromosome aberration, we know the chromosome aberration has number aberration and structure aberration. And in the number aberration, maybe a patient will have more than 1 or 2 X chromosomes. For example, in the male, the normal male, the sex chromosome type is XY, and the female is XX. But in the Turner syndrome, the sex chromosome type is X, only 1 X chromosome. And in the Klinefelter syndrome, the sex chromosome type is XXY. And in the super female syndrome, the sex chromosome type is XXX. So according to the number of the X chromosome, we can see in the normal male, it is 0 Barr bodies. In the female, it's 1 Barr bodies. And in the Klinefelter syndrome is 1 because he has 3 X chromosomes, so he has 2 Barr bodies. So we can detect the abnormal the number of sex chromosomes. Mm, if we learn how to observe the body, first we must know body, and then we learn some other knowledge of the body. But first we will know how to prepare a specimen for us to observe. But if we have the cell specimen on a slide, we will observe them under a microscope. But how can we know which is the body? So the body must have some characteristics. Then first, generally the bodies occurs in the interface of the cell cycle. So, and do you still remember the composition of the cell cycle? The cell cycle has the interface and the anaphase, and the interface occupies the most time of the cell cycle. So, in this condition, we have many opportunities to observe the body. But because maybe some other cells will during the mitosis phase of the cell cycle, so maybe we will not even in a normal female cell, we will not observe the body. During to the scientific results, about 1/3，就是about 30% of the female cells will observe the body, and the diameter is about 1 unimeter. This we can't measure it because the microscope in our lab have no the measurement machine or the have no the rulers and in a microscope. And here the body generally located in the edge of the inner nuclear membrane. Here you should pay attention. So the body is located in the edge of the inner nuclear membrane because the nuclear membrane have two layers, the outer membrane and the inner nuclear membrane, and the body located in the edge of the inner nuclear membrane. And finally, you should observation may be different depending on the material and must be you should distinguish the body from the nuclear line. The nuclear line are often far from the nuclear membrane or near the center of the nucleus. So in the picture from on our PPT, this PPT, we may be here. I didn't use an arrow to point the bar body, but here is a bar body with a condensed dye. But sorry, because this so this experiment is by Chinese students, and I forget to get more photos or videos for you, because this experiment is um, um, relatively simple for you, because many other experiments may be difficult for you to observe. So So I didn't take too many pictures and videos. And the experiment material is epicycle cells of oral and abdominal mosquito of males and females. And the experiment equipment has a microscopy, slider, the car glass, and a sterile toothpick. Because we should to get the epicycle cells of a male or female from the mouth, so from oral. So we need the sterile toothpick. And the most important experiment regions is the gym cell staining to see the body fast. The experimental steps is first you should wash your mouth, then use a sterile toothpick to get your epicycle cells, but discard the first scrap. You should patient. Use a toothpick, or you should gently to scrub your mouth. Like the picture, the area is shown in our PPT in the picture. Then you apply the scrap onto the slide, then dry it in the air for about 5 minutes is okay. Then we use the ginseng dye solution to dye the cells for about 10 to 50 minutes. Here, you should attention. Do not let the dye solution dry. You should observe it carefully. Then after dye the cells, then pour away the dye solution, rinse with water. Wait for then dry the slide. Then cover the slide and put it under a microscope for observation. So this experiment steps is relatively simple, but you still have some steps to pay attention to. First, describe the muscle membrane of the oral and barrel areas. Then you should do not with too many power, but you should also use some power, because if you just tap the mouse, then you will not get the cells, but some other materials or some dye cells. But if you with too you scrap the mouse with too many strings, then you will hurt yourself. So here, then when you apply the scribe onto a slide, you should put it as stretch with even with even a thin layer, then you can observe the cell with a single layer. Then it is easy for you to observe the body. And for the staining step, you should test a gradient of the solution of staining time. If you stain the cells too short, then you will not observe the dye cells. But if you stain the cell with the solution for a long time, then all the cells will die with deep dye. So you will also not observe the body. So here you should pay attention. Even though these steps are simple, but that have some steps or some tips for you to pay attention to. Here's a notch. So there must be scrap the cells. At the same time, should not be too long. Otherwise, the nucleus will be deep colored and its chromatin will be difficult to distinguish. Here, the last notes. Is the topically countable cells were detected under the low magnification. And the criteria was as follows: no bacterial contamination around the cells, clear cell counter and clear nuclear membrane, and no nuclear defect. So finally, I took pictures for you with this three. Then the last, because as this criteria the last picture shows the Barr body perfect, but this cell is not the clear cell counter and clear nuclear membrane. So I just show you a part of this picture. Then here, the center, in the center of the cell is the nuclear. Then, because the chromosome or chromatids located in the nuclear, so we should observe the nuclear for the X chromatin, the Barr body. So if you carefully observe this picture, then you can. This picture, so the first picture, so several points with deep dye, but only one is the Barr body, because other maybe some other materials that here maybe some bacterial contamination around the cell. So here this one is the Barr body, and in the second picture, the Barr body is not easy for you to observe. Here is around here is the Barr body, and in the second, in the third picture, here is the Barr body. So the assignment for you is to select one picture and then point out the Barr body. This is the first experiment. Second is the preparation and observation of human chromosome. Here you should we have learned the preparation of human chromosome and the band chromosome in our theory class, in the chromosome class. But how to make a chromosome space of human in detail, then we will learn in our second experiment. So the purpose for this experiment is for you is to master the preparation of chromosome specimens. And we will observe the unbanded chromosome, because we will use the gemstone or the lipid to stain the chromosome specimen. As we learn in our chromosome variation class, we know many chromosome variation can be observed under a microscope. But and you should describe a karyotype of a chromosome of a person. But first of all, you should make a chromosome specimen of this person. Then you can detect his composition of the chromosome and some other abnormalities of his chromosome. So this experiment for you is to master the preparation of chromosome specimen. It is a classical method. Maybe you can search on the internet. Maybe some other method also can be done for you to prepare the chromosome. All this method is okay, because our final goal of the is to get the chromosome space of a person. All these methods we talk about in our experiment is some classical methods for us. But with the development of the scientific technology, maybe some other method can be also maybe will be more simple or more convenient or more simple to operate for you. That is also okay, but you should know the not also the technology, but also the principle. So first, we will learn the principle. After short-term culture, the cells in the exponential growth stage can be blocked in the metaphase with Chromosome treatment. Then here you should pay attention. Why in the metaphase? This also so you need to know some knowledge of the cell biology. Because the chromosome in the metaphase, it is located on the center of the cell, and it can be observed and its morphology is the best for us to observe in the metaphase. So we need many cells in the metaphase to do our experiment. To get the metaphase cell, we need a chemical reagent, the Chromosome. Later, I will show you why this reagent can get a metaphase cell. Then, through the hypertonicity, the friction, drip, and staining, a large number of cells in the metaphase can be obtained, and the chromosomes can be clearly observed after making a class slide of the chromosomes with. So here, first, in this experiment, first you should get the metaphase cells. Then you under hypertonicity and fixing and drip and thinning, then we will observe the chromosome. And our experimental material is a Hacker 293T cells. It is a human embryonic kidney cells. And the experimental equipment supplies as a temperature incubator or a incubator bars, the centrifuge, microscopic glass slide cover glass, and the reagents for us is the Chromosome, the low osmotic solution, the fixed solution, methane and glacial acetic acid, and the depidite. And the low osmotic solution is 0.075 mol per liter potassium chloride. And then later we will learn the experimental materials, because we want to observe the human chromosome, so we should use a human cell. In many experiments, it uses papillary bladder cells, but we have no papillary cell, so we use some cultured human cells. And the HEC293 T cells is the common cells that we culture in our lab, so we use this kind of cells. But many other cells is also okay. And we use these reagents. So the function of these reagents, then we will learn their function. The Chromosome, it can prevent the microtube from assembling, then destroys the structure and blocks cell mitosis. So this reagent can block the cell in the metaphase. Then we will a large number of metaphase cells, and the later the potassium chloride solution. It by means of the reverse osmosis, the cell expands and the chromosome are spread out to observe the morphology of the chromosome. So it is a low permeability solution. And then if the chromosomes are released from the into the cytoplasm or from the nucleus, then we should fix their morphology. Then for us to observe them better, then we use a fictive named the canofictive. The canofictive is here. The menser versus the glycol acetic acid with a volume 3 to 1. So is this fictive named canofictive? It can rapidly penetrate the cell, implanting it and maintain the structural integrity of the chromosome. And finally, the DPI, it is a DNA dye. It can make the chromosome with a blue-purple fluency. So we use this in our first experiment. We use a giemsa. Also, in this experiment, we can also use the giemsa to dye the chromosome. But in this experiment, I use the DP. I also use the DP and giemsa. But in the giemsa experiment, I didn't observe the a better morphology of the chromosome for us. So, but in the DP group, I take a picture with the ideal result. So I show you the DP dye. But if we use the DP dye, we must observe the slide and a fluorescent microscopy. Then is the experimental steps. This first I will sorry for you that because this experiment will continue for about 3 hours. So I didn't take a good video for you. Then I just take some pictures. But later I will show you a link for you. Then you can learn some other knowledge of this experiment on the internet. So For simple, the experimental steps is first is the cell culture, as our picture show you. Then we will treat the cell with a chloroquine for about 15 hours. Then we collect the cells, and the cell will through the low variability treatment, then fix the chromosome and finally drop the side, then dye the chromosome. Then we use the microscope to observe the chromosome. First picture shows the culture box, the cell culture and natural condition. The second picture shows you the cell morphology and a microscopy. And then circle one shows some metaphysical cell, because the metaphysical is a mitosis cell, so they will not touch the box tightly. Then we will become maybe round the circle of the rounder morphology. Then here shows the detailed steps. First, we collect the metaphysical cells. Then we wash the cell with PBS once and digest with trypsin, and then add the DMEM culture with 10% FPS to terminate the digestion. Then the cell will blown off centrifuges at 1,500 for 5 minutes, and then the supernatant was discarded. Next is the low permeability steps. We use the potassium chloride solution with 6 milliliter. Then put them at the 37 centigrade for sorry, here I for this. The potassium chloride solution will pre-warm at 37 centigrade. Then put it to the cells and gently glue the cell with a paper tube for about 1 minute. Then mix the cells and incubate them in a 37 degree water bath for 20 minutes. Then here you should pay attention. After the water bath, you should drop the effective immediately, about 2 milliliter, into the centrifugal tube. Then blow it gently with for about 1 minute. Then centrifuge at 1,000 circle for 8 minutes. Discard the supernatant. Then we fix the chromosome again at the newly prepared fix. Here you should pay attention. The fictive solution is fresh prepared. So Maybe the potassium chloride solution, you can prepare it before the experiment, but the fictive solution you should newly prepared during your experiment. Then we fix the chromosome at room temperature for 30 minutes, then centrifuge it under at 1000 circle for 8 minutes and discard the supernatant. Then we get the chromosome, but how to observe it? So we should put them on a slide. But here you should pay attention. This is Esther slide. So we put the slide at a refrigerator ahead of time to make the slider cool. So here it named drop Esther slide. Because with Esther slider, then with the temperature difference, because the chromosome solution is under room temperature and the slide is Esther. So with the temperature difference, the chromosome is more easily to released from the nucleus. According to the number of the precipitation cells at appropriate amount of fresh fictive, less than 500 uniliter. Gently blow to make cells suspension. Draw a small amount of cell suspension with an eyedropper. Drop it onto a slide with pre-ice bath. Blow gently and dry. Finally, we use a depth solution. The 2.5 unit gross per milliliter depth solution to thin the chromosome for 20 minutes. Then rinse with water, blow dry with a high driver, or put it under the temperature. Wait for about 10 minutes. Then we use a microscopic, a fluorescent microscope to observe the chromosome. So from here, from the detail steps, you can see here we will last about expand about 10 minutes. Then here is about half an hour. Then here is about 10 minutes. Then here about 40 minutes. Then here with the drop slider and the dry procedure and steam. Here will about 40 minutes. So this experiment will last about 3 hours. Actually, for an ideal result, maybe we can see the chromosome like this. Maybe here has a nucleus because the chromosome are released from the nucleus. But actually, this picture, sorry, I took the picture like this. Here, the circle one is the nucleus, and then this is the chromosome. But because we use but our microscopy, fluorescent microscopy, has no 100 magnification, so I use a 40 magnification to observe the chromosome. So this picture, you can see the chromosome, but can't see them clearly. So we can't use this picture to number the chromosome, but we can use this picture to show our experimental results that we use this method to make a successful chromosome specimen. So the assessment for this experimental is you should show your result. This for you use only this picture because this experiment is relatively difficult for us. I do about 5 times, but only this picture is the best. So I'll show you the best. If you have the chance to do the experiment in our lab, then I will teach you how to do this experiment again. Then here you should so here is you should notice that I didn't give you the matters need attention in the experiment. So you should summarize the matters need attention in the chromosome preparation. And here, but with these regions or some detailed steps, you can have some ideas. Maybe here the slide is either or after permeability, you should have the prefixation steps, then centrifuge. I have another fixation step. So all this is the matters that's for you to pay attention to. So here, this is all the this is all the content for our today's class. If you have any question, you can ask me on the class or after class. Do you have any question? Okay, later I will send all the PPT and to you. Then this is the final class for you for the medical genetics. And then later I will send you all the PPT and then the summary PPT for you. But today I talk about 2 experiments. So the report for you, you can send the report to me before sorry. Before June 5. Okay?
OK 老师
Hello.
OK 老师 OK 老师
If you have any question, you can ask me.
OK 老师 OK 老师 OK 老师 谢老师，拜拜。
老师，
拜拜。
老师，thank you。老师，should we type the report or write by hand?
What?
Should we write with handwriting or we can just type in Word?
Sorry, I can't hear you clearly. Maybe you can send the message for me.
OK，老师。
谢谢老师，再见。